Welcome to today's Commonwealth Club World Affairs program. I am Quentin Hardy, an independent journalist and your moderator for today. The club would like to thank Wonderfest for being the community partner for this program. And it will be my pleasure today to introduce and speak with our guest, Carl Zimmer, author of Airborne, The Hidden History of the Life We Breathe. Carl is a respected science journalist and writes the Origins column for the New York Times. He is a 3-time winner of the American Association for the Advancement of Science Journalism's award during the COVID-19 pandemic, contributed to coverage that won the New York Times Pulitzer Prize for Public Service. He is an adjunct professor of Yale University and has the distinct honor of having an asteroid and a species of tapeworm named after him. Apropos of the tapeworm, if you have not read Carl's book, Parasite Rex, it's a charmer. Before we get started, if you have questions for Carl, please submit them in the YouTube chat and we will get to them later in the program. Carl, welcome back to the club and congratulations on your latest book.
Thanks, it's good to be back.
Great to have you. You know, the initial thing that struck me about this book, and I kind of think it motivated some of your work, was the way that people take the land and the water as habitations for living things. But they regard the air as this neutral, indifferent space we all move through. It has no deep relevance for the biological phenomena on Earth. But in fact, you show it's anything but. Why don't we start by having you talk a bit about that?
Yeah, I think that it's natural for us to kind of treat the air as this invisible, transparent medium that doesn't really matter to us. Of course, we know that it's where the weather happens. So we get cold, we get hot, we get rained on, and all of that.
You have to breathe something,
and that's right. And we are breathing. I think the problem is we're breathing all the time, thousands of times a day, so that we don't even think about that. And we might see a bird flying overhead, but we don't really think about, well, what else is flying overhead? And in fact, the reality is that it's a vast ecosystem that goes all the way up to the stratosphere and, affects not only our health, but really the whole balance of nature.
Now, you use a term that was new to me and maybe new to many of the viewers today, which was aerobiology. And I've since learned another one, aeroecology, the interactions of the biology in the air. How long have people been thinking about the air as a living environment?
It's interesting because, you have to then get into these kind of weird questions about, well, what do people mean by life? I would say that, for, say, ancient Greeks like Hippocrates, for example, they saw the air certainly as something that could cause diseases. And they would talk about miasmas, but these would be kind of, corruptions of the air, disturbances of the air, which would then waft along and people would inhale the miasmas and get sick. Then I would say, even in the 1500s, there were these fascinating ideas from, an Italian doctor named Fracastoro he envisioned seeds of disease. He was really one of the first people to envision a real contagious theory of disease. And he imagined that seeds could come out of someone's sick body and they could stick to their clothing or to other surfaces, but that they could also float through the air. And so he was picturing that, but this was long before anybody actually had seen, a microbe through a microscope. So it was just this almost like a feat of the imagination. Really, in terms of what I'm talking about in my book, that the air is full of life, full of separate organisms. It's actually not really until the 1800s that this starts to emerge. And there are people who are developing this idea that I think a lot of people will have heard of but not realize this is what they did. So like Louis Pasteur, for example, he developed this idea that the air was full of what he called floating germs. And he got into a debate with a leading scientist at the time who could not tolerate this. This was absurd. And there's a quote from a journalist at the time, this is 1860, who said to Pasteur, the world into which you want to lead us is too fantastic to believe. So it's been a hard idea to wrap our heads around for a long time.
And it's an interesting tension that keeps coming up, and we can get into the whole germs versus miasma, how big this stuff can be, little droplets, aerosols, many tensions that in your book culminate in COVID, and we'll go on from there. And I want to get into that in just a minute, but one of the things that came up for me was the kind of excitement people had as they discovered, there are spores in the air, and those little dandelion things that are floating in the air are seeds, and that's contributing to air, but it goes up for miles and miles, and it's partly living things, or it's bits of insects, or it's stuff collecting on or falling off of flying creatures. It's life in clouds. The air is in many ways a vector, a means of transport, but in some cases it is really its own ecological system, in a cloud, for example, or in an exchange with a plant. So can you speak about that a little bit?
It's a challenging thing to think about life as you talk about it as an ecosystem up there. When some of the first discoveries were made, I write about this in the book. In the 1930s, Charles Lindbergh, for example, took a probe with him when he and Anne Lindbergh were flying over Greenland to Europe in 1934. And it was shocking that he was catching stuff, miles in the air in over this Arctic wilderness. People were astonished. This was literally like front page news all over the place. The probe that he had built and the whole experiment had been set up by someone named Fred Meyer, who sadly has been forgotten. He actually coined the term aerobiology, which you mentioned before. And then he would go and get other people to do these experiments, like Amelia Earhart or these balloonists who went up into the stratosphere, some of the first people to go up into the stratosphere.
I was terrified at some of these experiments.
Oh, yeah.
30,000 feet up, 70,000 feet up, and the balloon starts tearing apart.
Yeah.
And they're worried about, okay, how can we keep our experiment going?
Yeah, that's right. This balloon called the Explorer from the National Geographic Society. Yeah, it went all the way up to the stratosphere, and they're having a grand old time. And then they hear a clunk, and they look up, and they're like, "Oh dear, our balloon is ripping."
That's not good.
Not good. And Fred Meyer had his probes on there, and to see if life could survive up there. They sent up spores and things, and so the crew is just three people in a metal ball basically, and now they're falling towards Earth. And yes, indeed, basically they said, well, okay, we're just going to manage our fall. We're just going to make sure everything's okay. We're going to kind of slow this down as much as we can. And, and they literally didn't want to jump off of this gondola until they were maybe, a mile, I think, or less from the surface, because they didn't want to land too far from the gondola. They wanted to land, and then they wanted to walk over and check out the wreckage. And they did. It's amazing. And they actually found Fred Meyer's probe, and lo and behold, it looks like these spores were able to survive. So incredible stuff that was happening. And, it's too bad that it's forgotten, because like at the time, people, again, more headline news. There was a headline in the New York Times: plant disease is 13 miles, in the air. It really challenged people to say, what is the upper limit of life? How high can life go? Could life just then go to other planets? And so all these things really first, came together in the 1930s, partly because of the technology that was finally available.
Yes. And taking it back to Earth, as it were. And this is an interesting tension point later on. The human interaction with the animal life around us is astonishing. I'll read briefly from page 396. Every day, we inhale millions of organisms, the vast majority of which do no harm to us at all. If the day happens to be in the spring, we take in thousands of pollen grains and tens of thousands of fungal spores. We breathe in hundreds of species of bacteria and viruses. You know, it's a whole other layer on. Nowadays we talk about the microbiome, but there also is this kind of aerobiome around us. These mixtures, these interactions of gases and liquids and smaller living things in which we participate unknowingly, just continually.
That's right. And it's coming, and it's, where are these things coming from? Well, they that pollen might be coming from a tree miles away. You might be breathing in algae that rose from the ocean thousands of miles away. You might be breathing in, a fungus that came from a wildfire in another country. There are all these sources for the aerobiome. Life is rising up into the aerobiome every moment of every day. And then things are traveling, following, the winds and so on. And then eventually coming down. Everything has to come down sooner or later. And, maybe you breathe it in, or, maybe it lands on the ground, or maybe, some animal breathes it in.
Or you exhale it and give it another quarter mile.
That's right. Exactly. We are, yes. And so when we exhale, we are not, our exhaled breath is not sterile, because our airways, our mouths, our noses are full of living things. They're coated with them, viruses, bacteria, fungi, so on. Like they live in our airway as well as our gut and on our skin. And so we exhale it, and primarily they're floating along in these tiny little droplets that we are exhaling. I'm exhaling them right now while I talk to you.
Yes. And of course, for the most part, they are benign and some are necessary.
Yeah, I mean, it's some might actually even be beneficial, and that's an idea that I explore at the end of the book, because we already know that there are many species of bacteria and maybe viruses too in our guts that we are benefiting from. They are probably shielding us from infection. They are sort of teaching our immune systems to be vigilant but tolerant. And there are all sorts of benefits to that. And then we also have this intake from the aerobiome, which might be sort of doing similar things in our airway and in our lungs. And there's some really interesting studies looking at what happens when people breathe in air in, say, forests as opposed to, cities full of blacktop. You're breathing in a different ecosystem. And it might be better to be breathing in more of that fresh air,
right?
Not just because it doesn't have pollution in it, but maybe because it has a better blend of species in it.
No, you might say it's the most continual participation with our environment we have.
Absolutely, yeah.
But of course, a big part of the book and a big part of the research for centuries now has been disease, and the air as a disease vector. And it was interesting to me that the people talking about the air as a disease vector in all sorts of ways continuously run up against the fiercest opposition of people. It's almost like there's a kind of bias toward the more heavily physical, like the earth is the culprit, or the ocean is the culprit, or things we touch are the culprit. This invisible stuff can't be the culprit. And this tension starts in the 19th century with cholera. Cholera is miasma. Cholera cannot be airborne. How do you account for that? Because the people proving this always seem to come up against opposition, no matter what the context.
Yeah, it is fascinating. I mean, because, we're kind of used to reading about, the history of science as sort of like a string of successes, as revelations, but then so and so discovered this and then we all agreed on that. And that's not really how science works. And actually there are some cases where it just seems like you look at things and you say, wait a minute, like why did people not accept this? Like, or why did they dismiss it out of hand? Like, and over and over and over again. And it is a fascinating exercise because, like, no one will say like, well, this is why I refuse to believe this. So you have to pick up clues. And so I would say that, the fact is that the germ theory of disease was really set in opposition to old ideas that emerged out of this concept of miasmas. So you would have this idea like, oh, the earth, the air is disturbed and people breathe it in, and that's why they get sick. And then you have
It's almost the miasma is almost a kind of fear of the underworld. Something emerges from Hades and is making us sick in a sense, and then that evolves into a disease theory around it.
Right. Yeah. Yeah. But this had I should say, like, the miasma view really, it had, explanatory power, like, so that, in the early United States in the 1790s, Philadelphia is getting hit by yellow fever. And the leading doctor in the United States, Benjamin Rush, is like firmly explaining exactly how it is that corrupt air, the corruption is caused by a sack of coffee, a rotten coffee that's sitting on a pier in Philadelphia, and that air then spread through the city and caused the yellow fever epidemic. Like, it felt right, just by looking at how the disease was spreading. So then you have the germ theory of disease, where they're working really hard to make their case like, no, it's not these miasmas, it's these germs. And it just so happened that some of the most compelling examples for this involved things that were not airborne. In other words, you mentioned cholera. Cholera is spread in contaminated water. It's a spread through feces, and they contaminate water and people drink it, or, it's in their food, and that's how they get sick with that. Typhoid was another one. Again, another thing where, like, sewage gets into drinking water, you can get typhoid. And so, it was so compelling. And again and again and again, you would have all these compelling stories about individual diseases. Yellow fever, not miasma, a virus inside mosquitoes, and on and on and on and on. Like malaria too, like the name literally like evokes its old idea about bad air.
Yeah.
Right. Bad mosquitoes with bad protozoans inside of them. That's what it is, really is. And so, so you are left in the early 1900s with leading public health people saying like, well, I guess we can set those miasmas aside. In other words, the air is basically harmless. I mean, they just
It's neutral.
dismissed. Yeah. They just didn't believe that germs would come out of people's noses and mouths to float away. Maybe if someone coughed at you and you got smacked in the face, sure, that's fine. But then you just need to step away a few feet.
That feels more like a physical event. That leads to sort of the large droplet theory later on, and we'll get to that with COVID. But another issue here is, let's just say it's the advancement of science also has a lot of ego involved.
Oh yeah.
And you get very strong egos, and you get heroes. Louis Pasteur is an authentic hero. He's a persuasive person. And while Fred Meyer is, gets the headlines and associates himself with heroes, unfortunately, some of the people who are correct in this weren't the easiest people to get along with. And
absolutely.
That's just a human irony. They may have been right, but they weren't great at selling themselves, as it were.
Yeah. So what the example that comes to my mind when you talk about that is this husband and wife team, William and Mildred Wells.
Exhibit A.
Yeah. I remember during the COVID pandemic, when I was puzzling, like, why is there all this heated debate over whether COVID is airborne or not? Like, shouldn't this be something that gets sorted out pretty quickly? I just had a lot to learn. And part of what scientists were telling me is like, you need to learn a little history, and you need to learn about the Welleses, who I'd never heard of, honestly, before COVID. And so they, in the 1930s and 40s, they just went full on against that view that the germs were just in water or food or close contact. They really were making a passionate and well justified case for airborne infection for some diseases. And they did a whole series of really remarkable experiments. And when I looked into like who they were, and this involves going into archives, finding unpublished manuscripts, finding letters, just everything I could find, because there's no biographies written of them. And I sort of reconstructed like who they were from all of this material. And yeah, literally a colleague of theirs, a friend, said they are their own worst enemies.
Yeah.
They had a really hard time communicating with people. They were really combative. Part of it was they were outsiders. William Wells had gotten this education at MIT as studying water and decontaminating water, and that was his thing. And then he got into like oysters and keeping oysters healthy and so on. And he ends up at Harvard just as a very poorly paid lecturer, and he wants to be a great scientist. And I think everyone's looking at him like, who are you? And his wife, Mildred, she had a medical degree, which was really unusual, especially, from Texas in 1915. I mean, so she was fierce and strong, but like also kind of taught herself epidemiology. And so, people at Harvard didn't know what to do with them. They after a while, like they made all these incredible discoveries in the space of a few years. They, again, were headline news, incredibly famous, and then Harvard just got rid of them. They were like, we can't deal with these people. They're just too much. Go away. And so, after that, they, were scrambling for years to try to survive.
What struck me in the Wells story also was, he started out doing water, but he was kind of haunted by the 1918 pandemic, what we call the Spanish flu, which we now think of as an H1N1 variant that was incredibly virulent, killed more people than the preceding First World War in a very short time, and then kind of went away and had its own airborne drama of masks versus not masking and with the vectors. I was surprised that that flu didn't, and the success of masks or lack thereof didn't affect aerobiology more. Just seemed to arise and vanish again.
Yes. So the 1918 pandemic hits probably starting with American soldiers, maybe at a military base in Kansas, and then spreads. The soldiers themselves who are infected are spreading it around on transport ships and so on. It's all over the place on the battlefront. More soldiers are actually sent to the hospital with the flu than are being wounded in battle. And here's William Wells. Like, he is actually hired as a sanitarian, as someone who's going to keep people safe, soldiers safe, by cleaning their water. He's taking the germ theory of disease to the battle lines, and he does a great job of it. But all around him, people are dying. from this flu, which is not waterborne. And I would guess that he had an inkling, like maybe this is airborne, but he didn't really have his ideas clear in his head yet about these things. And but it was very traumatizing for him. After the soldiers got it, it went into the civilian population all over the world. 50 to 100 million people died. That's a lot when you think about.
I have to insert a personal story here.
Yeah.
When my grandmother was about 100 years old, she would have episodes of not so much dementia, but not being aware of where she was in her own life. Her memory was still good, but she was kind of floating across her own past. Yeah. And I came to see her one time, and she thought I was my father. And she talked about, my father was born in 1918, and she talked about what it was like when I was a little baby and there was this flu coming through killing everyone. Wow. And there was nothing you could do. Oh. And it was just the emotion doesn't resist time. Yeah. You could just sense what this was like in the world for everyone right then. When there was no path the way there was with COVID. It was just coming through and killing people, and you didn't have a clue. It was actually one of the more chilling experiences of what the world was like before antibiotics and disease control.
Yeah, I mean, one reason potentially that the flu pandemic was so bad was that once people got the flu and their lungs were weakened, then they would get bacterial infections coming in secondarily, and there were no antibiotics. So no antibiotics, no antivirals, no flu vaccine that actually worked. They thought they had a vaccine, but it was against the wrong thing. So yeah, it was a mess. And William Wells's sister died at age 27 stateside of it. So and I do think that he was quite haunted by it. And when he developed his ideas about airborne infection, he really believes, like, I think we can stop the next pandemic. He would say, like, now that we understand how these airborne diseases work and we know how to stop them in their tracks in midair, maybe we can bring the next pandemic to a halt.
Now, people sometimes have, as I say, unfortunate personal circumstances with institutions or in their own nature. He also wasn't a good writer. He couldn't communicate well, and that was a problem. Another difficulty in the course of accepting aerobiology has been its allies in the form of the weaponization of disease. And,
that's right.
There's some fascinating chapters on that. So,
I mean, it's an important sort of kind of dark side to this history because and quite a remarkable one because here you had, I mentioned, Fred Meyer is flying around in planes trying to understand, how it is that, fungal spores are spreading and how it is that they might be able to reach farms and wipe out their wheat fields. In the 1930s, this is going on. And then you have William Wells and Mildred Wells, who are actually learning about how airborne infection works, as in terms of spreading droplets from person to person and the dangers of indoor spaces that aren't ventilated, and also like a potential, way to treat them with ultraviolet light. And all these things are happening in the 30s, and then World War II comes. Fred Meyer, tragically dies in a flight when he's hoping to launch the science of aerobiology. I won't get into that too much, but then you have the Wellses who are, they see this as their chance to really demonstrate airborne infection and ways to control it on a huge scale. Like we'll fit out these barracks and we'll be able to protect people and so on. And because they're just not on the inside, they don't know, they're politically, are very ungifted, they're just shut out, and people do other research and so on. But secretly, unbeknownst to them, the military has decided, what we would like to do is we would, instead of protecting people from diseases in the air, we want to figure out how to make diseases in the air. We want to make biological weapons, and the best kind of biological weapons are airborne ones. And so they build Camp Detrick in Maryland, now known as Fort Detrick, and there they build something called the Aerobiology Building. And in there they actually build things that are sort of modified versions of what William Wells was building. And then they start like pumping in things like anthrax and other pathogens into these chambers to see how much do you need to kill a mouse or some other animal. And Wells didn't even find out about this till after the war, and he was shocked and horrified. He called it the suicide of bacteriology. He really did feel like this was a terrible, terrible thing for scientists to get involved in. And the fact is that the United States continued to build huge stocks of biological weapons well through the 1960s, tested them out, sometimes on people. The Soviet Union ended up being even more ambitious with their work, so that even after the United States and the Soviet Union signed a treaty in the 70s, the Soviet Union just basically pretended like they hadn't and kept secretly making this stuff.
Got to say, in 1982, I had the strange fortune to land on Johnson Atoll in between Guam and Honolulu, which was where they were storing a lot of anthrax. And the plane would land, and people who were allowed to work there would get on and off, and none of the rest of us were allowed to get off the plane. And it turned out later, yes, there were enormous stocks of biological weapons and other things out there.
Yeah. I mean, it's the legacy, of biological weapons is a really complicated one. Because, I mean, on the one hand, probably the best demonstrations that diseases can be airborne were actually carried out at places like Camp Detrick, but most of it was classified. So
I know, it's shocking. While the government officially, while health officials can't even bring themselves to think about airborne disease, the military is sort of adding up the kind of fantastic numbers they can take if they land one of these on Moscow.
Yeah, and I mean, you actually can find like individual people who are sort of saying these seemingly contradictory things, like Alexander Langmuir, who is very famous as really the first disease detective at the Center for Disease Control, like just a really towering figure of public health, did amazing things. He really, he looked at William Wells and just said, like, your ideas are just not working. I'm going to bury you. Like he helped to get his funding cut off. It was really quite something. And he really didn't, for a long time, he just didn't have any time for the idea of airborne infection. Meanwhile, he was actually, secretly going to Camp Detrick. He was consulting for the biological weapons program. And, the Center for Disease Control, I mean, really a lot of their disease detective work was based on the Cold War preparations for a Soviet attack. How would you know that we were under attack? Well, you send out these epidemiologists. So, I think maybe, Langmuir, in that case, he just believed like, well, I don't think that diseases can be naturally airborne, but, with a little human ingenuity, they can.
Right.
Which again is, like in hindsight, it's like, no, that's totally not true. And he actually himself later in life started to realize like, I think I was too harsh on Wells, but the damage was done.
Oh, yeah. Well, let's, we don't have a ton of time here, and I have some questions from the audience I'm going to get to, but let's move through the fabulous diseases of this century, SARS, H1N1, Ebola, and of course, COVID, which have all been kind of faithful to our understanding, our current, much more sophisticated understanding of the aerobiome. What have we learned from this series of events?
Well, I would actually say that we collectively haven't learned that much. The SARS epidemic in 2002, 2003, that was a coronavirus. And that was the first time that a coronavirus proved to be not just something that could give you a cold, but could kill you. And it had jumped from bats to intermediate mammals, and then it affected people in China, and then spread to places like Canada. And actually, there were people who were on the ground dealing with the SARS epidemic who, were kind of looking at how people were getting sick. And, the prevailing paradigm would have been like, well, you get it through contact, or, maybe, if someone is coughing directly at you, you could get it that way at short range. And they were saying, no, I think this is happening at a much longer range in some cases. It might even be like moving throughout whole apartment blocks. And so they had to rediscover William and Mildred Wells. I talked to Hugo Lee, a scientist in Hong Kong, and he's like, I kept seeing these references to this book by William Wells, and it was nowhere to be found in Hong Kong. So William Wells wrote one book in 1955 with his vision of things, and he had to get someone to photocopy it in a library in the United States and ship it to him. And then he started reading it, and he's like, oh, it's all here, right? And so, but even, when people like Hugo were able to start to put evidence together, we need to think about airborne infection, the prevailing views, the consensus, the public health guidance really did not change. And then other people, the other things would happen. You mentioned H1, the 2009 flu pandemic. That brought in other people. Lindsay Marr, for example, at Virginia Tech, who had been studying air pollution and sort of the physics of the atmosphere. Her kid was getting sick all the time and having to come home from daycare. And she's like, what is going on? And she got interested in William Wells too, and could see how the current guidance was all messed up. And so you had a few people as these various epidemics were happening, who would get involved in airborne infection research, and we're really trying to get other people to listen, and people were not listening. And so when COVID hit, you still had a lot of public health guidance and a lot of sort of assumptions about how diseases work that just were wrong. At least when it comes to COVID. I mean, we had this legacy of assumptions and bad ideas and misunderstandings and people getting forgotten and all of that, that all piled up into basically a situation where it really was a big factor in why we were so unprepared for this pandemic.
Yeah, let's spend a minute on that. That COVID, I'm sure you saw the video about how to unpack your groceries.
Sure.
All
I was wiping down my groceries, absolutely, because that's sort of what made sense. I would wash my hands, singing happy birthday twice. Now, washing hands is a good thing to do, and we all need to keep washing our hands. But, I remember going to have dinner with friends of ours, and this was in February, and I was warning people about the pandemic, and they thought I was crazy. And I was like, okay, I'll come to dinner with you, but I'm not going to give you a hug, and I'm not going to shake your hand. And I was I thought I was being sensible. But then, of course, then we sit down at the table and we talk for 2 or 3 hours. Just, If I had COVID, I could have killed them. But so in hindsight, it's amazing. But yes, I mean, certainly this was not just what people weren't wiping down groceries just randomly. People were being informed, this is one of the things you can do. Like on CNN, Sanjay Gupta is like, here you go, take your Cloroxes, wipe down your bags.
Yeah. Well, we'd all seen Contagion, and so we knew the word fomites, and we'd seen outbreaks, so we thought Ebola could jump to being an aerosol. There was a lot of pop science misinformation out there. I'm not sure that was the chief problem as much as, within the scientific community. Once again, this kind of bias to, okay, if it is airborne, it's in heavy droplets, so 6 feet is okay. Yeah. And again, it's almost a bias toward if it is going to be air, it has to be almost physical air. It has to be clumpy air. It has to be, it can't just be in tiny little aerosol droplets.
Yeah, yeah. I
Depends
mean,
how far.
I sometimes will bring, I don't know if I have it here. I sometimes bring a little carbon dioxide monitor to my talks.
You mentioned if it gets above 800, you leave the room.
Yeah, yeah, yeah. I, it must be in my other room, but I'm just checking, see if I can find it to show it off. But yeah, so it's just this little monitor, about that big, and it just has a number showing you the concentration of carbon dioxide in the room. And if a group of people just get together in a room, you might start at like 600 parts per million, and then it just goes up. It'll go up and go up and go up. Why is it going up? Because we are exhaling a lot of carbon dioxide in our breath, and we are sort of replacing the fresh air with what was in our lungs. Now, people don't like to think about that, but we're just doing it all the time. All the time. That's just what we do. And again, that measures carbon dioxide. We don't have any good way to measure the living things that we are exhaling, but that's a proxy. That tells you, like, okay, this room is filling up with our carbon dioxide, and it's filling up with our droplets, some of which are going to have things living inside them. And if you're in a pandemic, some of those things might make you very sick. Yeah, yeah.
Yeah, and I enjoyed the insight that the diseases coming, the life coming from the deepest recesses of our lungs, which is likely to be the action zone for disease buildup, are also likely to be on the smallest particles as we exhale.
Yeah, yeah. It turns out that there's this whole kind of fascinating world of physics about our breath, and so as we exhale, there are lots of different ways in which tiny little droplets can form and get released. So, like the vibration of my vocal folds, for example, will produce droplets of a certain size. And then in your lungs, you have these tiny passageways, and sometimes they will stick together, and then they will pop out again, and they'll produce droplets. And then those droplets, as we exhale, will make their way all the way out from the recesses of our lungs and out of our nose and mouth. And those will be very small, those ones that are down in there. So it's definitely true for certain diseases that only tiny droplets can get in there. So, for example, tuberculosis, which William Wells worked on, it was the last great disease that he tried to prove was airborne, and he died trying to do it. And so the bacteria that causes tuberculosis, they only grow once they get to the tips of your lungs. Like, that's it. They can't grow anywhere else. So it's a very odd, particular way of making a living, but that's what they do, and they kill 1 million people a year, so they do it really well.
Yeah, success.
Yeah, they're an incredible success, and and so only the tiniest droplets will be able to get in all the way there, but that became the source of a lot of confusion, because then, public health authorities, Center for Disease Control, probably Alexander Langmuir himself got confused about this. People started to say, like, well, airborne diseases can only be spread by droplets that are smaller than 5 microns. And then that got sort of confused, and people said, well, things that are bigger than 5 microns can't float. And it's like, and so a physicist like Lindsay Marr would say, that's crazy. Where are you getting your math from? That's not true. So in fact, droplets of lots of different sizes can float. Part of the reason for that is that, some of them are getting kicked out in a plume that gives it an extra push. They're also evaporating, so they're actually getting smaller. And then once they get small enough, they can float. So
5 microns turns out to be kind of a science meme. Like, it's an arbitrary pick.
Right. But that was in, all of the standard public health guidance at the start of the COVID pandemic. You would just come across 5 microns all over the place. And so, and this was even after people like Lindsay Marr had been trying to get people to understand that that doesn't make any sense in terms of basic physics, and it just wouldn't change. And I think that maybe speaks, there are a lot of, fractures here, a lot of, failures to communicate. One of them, I think, is between different disciplines. So you have, public health people are much more focused on their patients and on epidemiological patterns, whereas you have someone like Lindsay Marr or Hugo Lee, who are much more into physics and engineering, and chemistry. And it's almost like they're kind of talking different languages, and so
layer on that the enormous budgets that are geared to execute along certain received lines.
Yeah. Right. Yeah. I mean,
it's hard to get off the path.
Sure. And if you are going to, try to make a scientific career for yourself, it can be very sensible to just say, I'm just going to kind of build on what everybody else has been doing and, make my increment and then I'll be okay. Whereas, I mean, you can see what happened to William and Mildred Wells. I mean, if you have a, yeah, it is kind of academic suicide.
Yeah. Well, I'll make you hand a report card in. How well handled was COVID, given what people knew, given what was done? What would we do differently seeing it now?
Well, it is striking that, I don't know. I mean, I think that, I think it might help to give, not just sort of, humanity a report card, but like particular countries' report cards, because, the United States had an incredibly, a very high death rate, despite being an incredibly rich country. If you look at the measures that were taken in places like South Korea and Japan, they were really like very much dealing with it as a serious threat, as an airborne threat, much more than the United States was. Certainly, much more than World Health Organization was in their guidelines at first. So in Japan they just said, like, it looks to us like these crowded places are really the places where this virus is spreading. So just try to avoid these crowded, unventilated spaces. That's it. Mm-hmm. That's it. And I think that went a long way. And they also had a much more comfortable relationship with masks, for a lot of cultural reasons, a lot of historical mm-hmm. reasons. Japan, China, Taiwan, Hong Kong, like they had a long history with masks where they didn't see it as being, a very sort of
their freedom didn't seem to be threatened.
Yes, they were not being triggered by masks the way that Americans were. That's
what did you make of the Swedish approach of just letting it rip?
I think that people have really, tried to use the experience in Sweden, for a lot of political purposes. I mean, it's not like they just said, let's let it rip. I mean, they certainly didn't have some of the more, severe restraints that some countries had, but they still had, plenty of rules in place, and, they were getting people vaccinated. I mean, and
it is a highly orderly society, and people agreed to common behaviors.
Yeah,
and it was backstopped by an excellent healthcare system.
Yeah, I would say though that it's we might have, as a world, we might have been able to stop COVID right at the very beginning when it was ripping around Wuhan. And even there, like sort of doctors on the ground were like, I think this is airborne. They would just say, like, I think it looks airborne to me. But, again, like the public health authorities were kind of giving out mixed messages and not really kind of like reckoning with the fact that it was airborne, and so they didn't really take measures that could have reined it in. I think at a certain point, this thing was just was so hard to stop after a while, especially as it was evolving. It was definitely, by the time you have Omicron, you have this form of the virus that can evade antibodies that we might have had from vaccination or infection. And some people have argued that actually it evolved to become better at being airborne. So you just had this thing that could just shoot around and just find people and get them sick. So that's, sometimes it's very easy for us to forget what happened in China. Finally, when President Xi decided he had to stop all of the COVID protections or he might lose power. Things were getting that bad. So when he kind of let all these things go, there's just staggering numbers of people got sick. Just a huge percentage of all of China got sick, and
Death.
Yeah, maybe 1 million, maybe 2 million people died in a month.
Mm-hmm.
I mean, we don't really know because they didn't want to track it or be.
We're not good at sharing.
No, that's absolutely right. That's absolutely right. But there are enough clues from the outside, people like Hugo Lee were able to infer, oh my gosh, this thing has ripped through this country, and a lot of its ability to rip through this country was probably due to its being extremely airborne.
I'll turn to questions in a moment. Two quick ones. One, do you have a position on the lab leak theory?
When you say lab leak theory, I'm always asking, what theory are you talking about exactly? What is the theory? Because some people will say, oh, there was something in this lab at this institute, and then other government agencies have been saying, no, we think it was over at the Center for Disease Control office in another part of Wuhan, and where they were storing things. And it's well, which is it? And what's your evidence for it happening there? So it's very difficult to evaluate a scientific claim that is being made by agencies that aren't going to share their information. That's not really the scientific way.
Right.
I mean, in my book, I write about an interesting, a definite lab leak, which was or a biological warfare factory leak in Siberia, where Sverdlovsk, where in 1979, I believe it was, they were making biological weapons, and something bad happened, and a bunch of spores blew out of the building. And they denied it. The Soviet Union denied that they were making biological weapons, and they said, "Oh, this is just a natural thing. It's just some cows, gave off
anthrax out there. We can do, yeah."
Right. But what then happened was that Matthew Meselson, a Harvard scientist, he finally was able, thanks to Boris Yeltsin kind of opening things up, to go and look over all the data and to kind of talk with people and kind of reconstruct what had happened. And it was very clear that there was, all the deaths, or most of the deaths, I should say, and the illnesses and the cows getting sick were in this straight line downwind from this particular place. And so, so there, if you present that, okay.
That's some evidence-based science, yeah.
Right. The people who are putting forward the lab leak, they have not done that. And I will certainly acknowledge that the Chinese government, as we just said, is very not forthcoming about this, and they've been criticized a lot about that. On the other hand, there have been other scientists who have been actually publishing in peer-reviewed journals a whole set of analyses, and they are arguing that it happened at a marketplace. That
Trans-species, wet market, that theory works too. Let's take it to a couple of questions. While doing research for the book, was there anything that particularly shocked or surprised you?
um, I
Probably a bunch.
yeah, no, there was a lot. There was a lot. Yeah, no, I would say that that was, I would just, as I was working on the book, I would periodically go to my wife and say, like, I can't believe I'm seeing this, or like, this is crazy. Yeah, and so it's hard to pick anything in particular. But like I would say that, digging further into the history of biological warfare was really something, the way that that the U.S. military decided one interesting way to see how biological weapons might be used on a coastal city was to send out a Navy ship and spray San Francisco with spores, which they did,
I took
and
that
then
personally.
they went and collected them, and they did it in New York City subways and all sorts of things like that, and that is pretty astonishing. But I do also think, like, when you go back and you look at some of these early the early work of people like William and Mildred Wells, where they we were sort of hashing things out during the pandemic in a very difficult way, and you can see that, like, they had kind of worked out a lot of the basics already, using very simple equipment, very simple ideas, but they got it. They even, in a measles outbreak in Philadelphia in 1940, they put ultraviolet lamps in a school there, and they protected their school from this huge wave of measles.
That is a very interesting thing you get into, ultraviolet light as a disinfectant, and something you see at the end at a COVID, an event where people are keeping careful about COVID, and you think
Yeah.
UV light could be useful in this as well.
Yeah, yeah. I mean, I'm not saying that the UV light is the only thing you can do to keep the air clean and to stay safe, but it's certainly one element. And it was the thing that William and Mildred Wells discovered, that it can, knock out influenza in the air, tuberculosis, and so on. And they really made a push to try to get ultraviolet light, used more widely. And there was a big interest in it for a while, but then, unfortunately, it's a sort of a theme of this book, things kind of petered out. But there are people today, scientists, who I write about, who are really working, trying to kind of find the best way to use very safe and effective ultraviolet light, just as little fixtures that would go in rooms where you can't get ventilation and so on. There are other things to do: filters, opening a window, air purifiers. There are lots of things that you can do to keep your air clean.
Is COVID over?
People are still getting COVID. I mean, no.
No, but is it kind of baked in now? The way, flu is not over. We live with flu.
Exactly. Yeah, yeah. And not only are we live with flu, but, like, there are the seasonal flu we're dealing with started as various pandemics. The 1918 flu pandemic, that flu and its descendants are still with us. Other pandemics where they were spilling over from animals, they're still with us with influenza. So COVID is still with us. It's not often that they go away. SARS, for example, seems to have vanished. And that was a real public health triumph. But it helped that with SARS, like, you were infectious when you were showing symptoms. Whereas with COVID, you can be pre-symptomatic or even not have any symptoms at all and still be able to exhale viruses and get other people sick. So I don't think we're ever getting that genie back in the bottle.
No, they evolve quickly and they adapt, and there's a lot of host material out there. We'll just have to bake it in. Couple of personal questions I wanted to get to. Of your 15 science books, aside from this excellent current one people should buy and read, what is your favorite?
What is my favorite? It's like asking.
What was the most fun to work on?
I guess, I guess my first book, At the Water's Edge, was, I was pretty young and I wrote a book and I was like, wow, you can do that. Like, you just write like crazy. You go visit scientists, you go bring stuff together, and voila, a book. And, So yeah, I suppose as sort of as a sentimental favorite, that's it. But you know, I've been really grateful to be able to write about all sorts of things over the years in these books, and the books really, you know, I'm grateful that I can, you know, that I can sort of step away from being just a journalist who is just focusing on what's happening right now. Because, you know, a new scientific paper comes out, it's not like the whole idea for it just popped out of nowhere from someone's head. Everything is building on history.
Yeah.
And that history itself is so interesting. Sometimes very frustrating, but sometimes very exciting too. So anyway, yeah.
I'll make a plug for Parasite Rex just because,
Okay.
well, the thing that did so nicely was um, really sharpen what makes reading these kind of books so great, which is you admire the ingenuity of nature and you admire the people who can figure out the ingenuity of people figuring out the ingenuity of nature. It's kind
Yeah.
of a,
Oh,
even when
absolutely.
it's scary as can be, even when these are utter monsters, it's like, that's amazing. It takes
Yes.
over a crab and changes its sex. Oh my
Right.
God.
Yeah. Yeah, and the pure sort of natural history of that kind of biology is can you just you sometimes you have to stop and go back because you're like, wait a minute, like I that sounds like a science fiction plot, and I'm sure I misread. Like, no, it's a science fiction plot. Oh my gosh, and it's happening like there on the beach. Oh my gosh, but you know, I would also say that one of the really fun things is to like to go and find the people who are studying those, you know, strange mind controlling barnacles or what have you, the people who are dedicating their lives to, you know,
Mhm.
parasites in all their weirdness, and they're just they're just so interesting to talk to, and they're really grateful that someone wants to talk to them about,
Obsessions are always
yeah,
interesting. It's the
absolutely,
delight in most journalism is portraying an obsession because it's like a kind of certainty. They're just so focused, you know,
yeah,
and the rest of us are so diffuse. And someone who can, in this book, someone who can invent this donut shaped object people stick their heads into and cough into it, it traces the size of droplets and gives a little bit more information to this quest to cure something. It's fascinating.
yeah.
Um,
You have to be kind of obsessive to gain this knowledge because it doesn't come easy, especially
yeah,
for aerobiology.
yeah, and you're up against the prevailing power and wisdom, and that's not always easy. As a you've been a long time science journalist. This is a question for the audience. What are some of the biggest challenges facing yourself and your fellow journalists?
Um, I think that I would say that, you know, certainly, you know, journalism in general is having a lot of, you know, commercial challenges, because for the kind of, you know, for the kind of work that goes into journalism, you know, people have to make a living, and there needs to be, like, you know, support for those, you know, investigative reporters who are going places to find the story. And you know, the old economic models, the ones that existed when I was just starting out in journalism, are gone. I mean, you know, classified ads, like that was a big deal. That sent a lot. You know, that classified ads paid for a lot of Pulitzers back in the
Oh
day.
yeah, 30
And
years. You don't have to tell me.
yeah, right,
And
exactly. I mean,
there's also a challenge now of you're a smart guy and you're at Yale. This stuff's getting harder to learn, isn't it?
it is getting harder to learn, and I do actually like have to sort of like budget in time just to get up to speed. So, you know, I might be writing about human evolution. You know, I had a conversation with someone just yesterday where I said, like, okay, the evolution of tool making. All right, here's my understanding of it as of like 5 years ago, and I know I'm falling behind. So help me
Right.
to understand, like, what do we understand now? What are the key things that have changed in that time? And you know, the scientist was very generous with his time, and now I'm up to date on that. But yeah, I'm just having to do that all the time because, well, at least for the time being, science is moving forward at a very fast clip.
Yes, and we now have computational systems that can view it in a series of interactions we couldn't see before, which is layering more and more complexity into it.
Yeah, the scope of data is amazing. So it was amazing enough that you could see all the genes that were active in a cell. Now there are databases that have, well, now there's actually a program underway right now to create a database of 1 billion cells with all of the genes that are active in them. And I mean, that kind of data, like you can't even as a scientist, really make sense of it on your own. You do actually need, computers. You probably need some kinds of good AI to help you with that.
I was going to ask how AI is figuring, in your view, in all this.
It's interesting. Like AI, I see it just a big contrast between AI within science, where scientists are using it and are being very skeptical about it because they want to be right, and AI that's being kind of like, just sort of thrown out there.
ChatGPT, chatbots.
Yeah. Like, here, let's give people something where they type in questions and they get answers that might or might not be right. Like, that's not what scientists want. And so if you look at the kind of some of the, I will say, like, there definitely is some AI hype within science, but then there are these demonstrations of what AI can do in terms of just, recognizing, say, new types of cells. Like they're just finding new types of cells. They're identifying new genes that were hidden away to our sort of human understanding, and that's good. But you can't then assume that, if it's AI, it does everything. Certainly not the case in the
the AlphaFold team. The AlphaFold team got the Nobel Prize legitimately, and that does show the good uses of AI.
It does.
But that was really good quality data, which you don't encounter in the normal world.
Yeah, but then people do forget that, like, these protein folding AIs, they also screw up. I mean, they do get things wrong sometimes, but we sort of focus on their triumphs, and we sort of forget that it's far from perfect. It's just a very powerful tool. And I just, we have this sort of instinct to kind of, bow down before computer overlords. We shouldn't.
Right. Good advice. Almost on that note, I do have a final question for you, and then we have to close today. How has writing this changed your own habits?
Well, I have my carbon dioxide meter that I carry around just to sort of like remind myself that, like, I'm this air is this common good that we share. I would say if I'm on a plane, I definitely put on a mask when I'm getting on a plane, and before the plane has taken off and the air is really circulating well through the filters. Yeah. Mm-hmm. But I mean, I was just looking at a paper where some scientists actually pulled the filters out from an airplane and said, "What's in here? Let's find all the genes." Oh boy, they found like hundreds of species of bacteria and viruses and whatnot. So there's definitely stuff sloshing around in planes.
Oh, Carl.
But okay. Hey, but but masks, masks are engineered in such a way that, that a lot of those particles will not make it through. And that's a good thing. I mean, is it a perfect guarantee? No, of course not. But certainly the consensus is from all these studies that it does reduce risk of infection. And hey, if I can reduce my risk, I'll go for it. So that would be one way. That'd be one way that I would do it. And I like to, open the windows more.
Yeah. Got another book in mind?
Not yet. I like to wait till inspiration strikes and then strikes again and then just won't let me go.
Find a real good question to ask for a couple of years.
Exactly. I'm waiting. I know it'll come.
All right. Well, Carl, it's just been a great pleasure, and I can't recommend your book enough to people. Every one of your books does this great service. It provides us with a new way of seeing. And having the world a little bit richer and the colors a little bit deeper is just such a delight. So
Well, thank you very much. That's
thank you
really
for all
nice.
the work on that. It matters a great deal. And our thanks to Carl Zimmer, New York Times columnist and author of Airborne, The Hidden History of the Life We Breathe. We encourage everyone to pick up a copy of Carl's book at your local bookstore. And if you would like to watch more programs or support the Commonwealth Club's efforts in making virtual and in-person programming, please visit commonwealthclub.org/events. I'm Quentin Hardy. Thank you and take care.